Questions about that? 'Cause this is the last piece of how neurons talk to each other. And it's the last piece of homework 7. All right, what is a synapse? A synapse is a little space from one neuron to another cell. And that cell could be another neuron, or it could be a membrane. So we really, instead of saying postsynaptic neuron, let's call it just a membrane. Oops. So it is communicating information from the one neuron to the next cell, whatever that cell is. That cell could be another neuron, it could be a gland, it could be a muscle. So it just depends on what that next cell is. So we have some terminology. The presynaptic neuron, I'll put write this on the board, it's important to that. So the presynaptic, so this is the one that's coming up upon the synapse. The presynaptic is the one that releases a neurotransmitter. It releases the neurotransmitter. The postsynaptic, I'm going to say membrane because it just depends. It could be a neuron, it could be a muscle cell, it could be gland. This is the one that has to have receptors for the neurotransmitter. I did a lot of self-abbreviations when I'm in school, several of which stay, and neurotransmitter is one, NT. Although we could say New Testament, because I did go to Oklahoma Baptist University. But in this case, it's neurotransmitter. So the presynaptic neuron releases a neurotransmitter. The postsynaptic neuron then has to have receptors for that neurotransmitter to respond. And that neurotransmitter binds to those receptors, and it tells that next cell to do something. Okay, that sets you up as to what's happening. So here we have motor neuron, cell body, dendrites, axon hillock, axon, wrapped in myelin sheath, nodes of Ranvier. And then we have the ends of other neurons. These are the axon terminals. Axon terminals, synaptic knobs, boutons. If you speak French, it means button. Because they do kind of look like little buttons on the end. That's the end that's releasing, okay, for the most part. It changes when we get to chapter 9. But the neurotransmitter is being released out of these little red endings, or out of these little blue endings. These little greenish colored endings. That's where the neurotransmitter is being released. So these are the presynaptic neurons, the red, the blue, the green. That's the postsynaptic neuron. Here's a picture of a synapse. So here's a close-up. Notice that this calls it the terminal bouton of the axon. So there we have the axon ending. And then right here is the postsynaptic membrane, which in this case is a skeletal muscle cell. It's also true for homework 7. The postsynaptic membrane is a skeletal muscle cell. Now, in the end of that neuron are these little vesicles, and these little vesicles have neurotransmitter in them. And so we'll talk about exactly how it releases it here in just a second. And here is an electron microscope photograph that's been artificially colored, showing the axons with the axon endings, and they are almost but not quite touching. And this is a postsynaptic neuron down here, so it's just another neuron. This is what it really looks like. Neurons are completely covered with synapses from other neurons. But we are just going to look at one for simplicity's sake. Now first we have an electrical synapse, which are in the central nervous system, and they are gap junctions. Remember that gap junctions, the cells share the same cytoplasm. And so what happens in one cell immediately happens in the next cell. Instead, we're going to look at the chemical synapses, in which the chemical is a neurotransmitter. And so now that stuff that you learned in chapter 6, now we need it, because now we're going to be talking about a chemical that binds to a receptor that causes ion channels to open, or causes a G protein coupled receptor to work so that ion channels will open. But now we can say specifically a chemical instead of a signal molecule, is what we had to say in chapter 6. Now it's neurotransmitter. So 2 parts. We have the end of the first neuron that's releasing the neurotransmitter, and then we have a receptor that's on the next membrane. And so the synaptic cleft is a little space filled with fluid. It's not very big, but it keeps nerve impulses moving one way. So this neuron, and then the next neuron, and then the next neuron, or whatever the next cell is. All right, step by step. So here is the end of the axon, and just to remind you of your anatomy, which I've already had you review, here's how I draw neurons, by the way. So there's the dendrite and the cell body, the axon hillock, the axon, and then here we have the axon terminals, or you could call them synaptic knobs, or you could call it a bouton. Many different terms for it. Money in the car. All right, here comes the action potential, zooming down this axon, and it's changed the membrane from minus 70 millivolts to plus 30 millivolts. Just reminding you what that action potential is looking like. And it gets to the end, and that's what we're looking at right here. This is the end. Close up of my little drawing. So here we have the bulb on the end. All right, there's that action potential. See, it's coming. And it's at plus 30. When it gets to the end, it opens voltage-gated calcium channels. So we've already talked about what a voltage-gated. Calcium channels open at plus 30. So as that membrane, it zips from minus 70 to plus 30 as that action potential, as that change in ions moving down, and it opens calcium channels. And calcium comes into the end of that axon. Then it makes these vesicles, these little green balls. These are vesicles at the end of the axon terminal that have the neurotransmitter in them. And it makes these little vesicles move down. It actually activates ATP. ATP is being used to do this. And it causes these vesicles to merge with the plasma membrane down here in the synaptic cleft. And so neurotransmitter, these little green balls, is dumped into the synapse. And it drifts across, because this is fluid filled. So it drifts across and it binds to the receptor that's in this postsynaptic membrane. Binds to that receptor and opens ion channels. And in this case, for your homework, this is a skeletal muscle cell. The ion channel it opens is sodium. Sodium now floods the muscle cell, which depolarizes the membrane. Already established that sodium coming in is going to make that membrane less negative, more positive. Then the neurotransmitter is broken apart, so it comes off of that receptor. The ion channel closes. The depolarization event ends. The muscle contracts. What we're doing. And then homework 11 picks up in the muscle. And then what happens in the muscle? So that's why we start homework 7 with a motor neuron. So you've now understood resting membrane potential in a motor neuron, the graded potential, the action potential, the synaptic transmission, which is the last part. So motor neuron, here comes the action potential, changes the membrane to plus 30, calcium comes in, calcium makes these vesicles of neurotransmitter, and this specific neurotransmitter, and it's a specific neurotransmitter, it's acetylcholine. It makes acetylcholine dump into the synaptic cleft, rifsacos binds to a nicotinic receptor. Acetylcholine has two different kinds of receptors, nicotinic and muscarinic. But nicotinic is in skeletal muscle. And that opens sodium channels, and that depolarizes the membrane. And then acetylcholine is broken apart. Sodium channel closes, and then that muscle cell is going to reset, repolarize.